it's 8 o'clock, so why don't we start it? Hello, everyone. So this is my great pleasure to welcome Dr. Adolfo Garcia-Ocana, who is giving a grant round seminar for our department this morning. He's an eminent scholar and a full professor on the faculty at Mount Sinai Diabetes, Obesity, and Metabolism Institute, and also he's in the division of Endocrinology, Diabetes, and Bone Disease over there. His research is broadly focused on diabetes, with attempts to develop new therapies in terms of preservation of pancreatic beta cells, their regeneration, and inducing immune tolerance to islet antigens. He has made throughout his career many important discoveries in the field of diabetes, and his work has been widely recognized and cited. So we obviously are very happy to have him today with us today. We wish it had all happened in person, but unfortunately, due to COVID, we have to do it online. So Dr. Garcia-Ocana obtained his PhD degree in Madrid, Spain, after which he moved to the United States, where after a postdoctoral fellowship in the Division of Endocrinology at Yale University, he moved to the University of Pittsburgh, where he rose through the ranks to associate professor, and then he moved more recently to Icahn School of Medicine in Mount Sinai, where he is now a full professor with tenure. Throughout his career, he has been very well funded. He received many awards from NIH and other funding agencies. He also has served on numerous occasions on grant review panels for NIDDK, the Juvenile Diabetes Research Foundation, American Diabetes Association, Medical Research Council at UK, and many other agencies. He also currently serves as a director of Human Islet and Adenovirus Core of the Einstein Sinai Diabetes Research Core. And this core plays really an important role because it provides other investigators with training in islet isolation, islet transplantation, and viral transduction of pancreatic islets. So, as I mentioned, Adolfo has made many important contributions in the field of diabetes, and he was first to discover the hepatocyte growth factor, AGF, that it stimulates proliferation of beta cells, survival of beta cells and their function, and that it improves beta cell mass in transgenic models, as well as it is beneficial in the setting of islet transplantation. Moreover, he and his laboratory has done a lot of signal transduction studies through which they identified protein kinase C-zeta as an important signaling molecule that is responsible for many of AGF effects on pancreatic islets. He and his lab is also involved in numerous collaborations. Perhaps one of the most prominent of those is that with Dr. Andrew Stewart, who is also at Mount Sinai. And this work led to this big discovery of DERK1A inhibitor called Harmine, which is a potent stimulator of proliferation of beta cells. And this is really important discovery because human beta cells are considered post-mitotic, and in adult person, pretty much they don't proliferate at all. So finding an agent that can actually overcome this obstacle holds a big, significant promise for therapy in diabetes. They published that finding on Harmine in Nature Medicine just several years ago, but that paper nevertheless received hundreds of citations since then. So this is really a really important discovery. However, in addition to islet biology studies, Adolfo's research has been focused on also on the aspects of immune tolerance and inflammation in the pancreatic islets, which I think is also very important because if you think about how to approach the problem of therapy in type 1 diabetes, you really have to develop a comprehensive approach which covers not only the well-being of islets in terms of how they proliferate or how they secrete insulin. But also, of course, you have to think about autoimmunity, suppressing autoimmune destructive response, as well as you have to take care of non-specific inflammation in the islets. And I think if you follow research of Adolfo, you can see that he tries to address all these problems. So Adolfo, thank you again for accepting my invitation and taking time this morning to join us. And I look forward to your talk entitled Regeneration and Preservation of Pancreatic Beta Cells for Diabetes.
Good morning, everybody. Thank you, Jan, for this very, very nice introduction and for the your kind invitation to give these ground rounds for the Department of Laboratory Medicine and Pathology. I would have loved to be there and to be present and meet all of you, even though probably it's freezing over there. But I would have loved to interact with you. But I mean, due to these difficult times, at least we have Zoom and we can talk to each other, see each other, and exchange knowledge actually this way. So I hope everybody's doing okay, everybody's feeling fine, and everybody's keeping safe. As Jan said, actually my work has been mostly on pancreatic beta cells and ways to preserve them, ways to increase their proliferation for regeneration. Lately, thanks to also my collaboration with Der Homan at the Diabetes, Obesity and Metabolism Institute and the Immunology Institute, have been able to start learning something about immunology, and I'm amateur, so bear with me on that particular area. Let me start just by saying something about type 1 diabetes, although everybody knows about this, but just for the sake of introducing my talk, it's a common chronic disease in childhood. There's almost 1 in 500 children in the United States actually is affected. And actually it's interesting that more and more adults as well are becoming type 1 positive people. Also, of those children with type 1 diabetes, only a small portion, 25%, actually achieve adequate glucose control. It's well known that type 1 diabetes has severe consequences, diabetic ketoacidosis, hypoglycemia, complications, multiple complications, and of course increased mortality. And it's almost 99 years since the first humans were treated with insulin as a treatment, not as a cure, to save them from a secure death. Actually, type 1 diabetes was death sentence actually in the early 1900s and before, of course. And this is just a CDC map of showing the incidence of type 1 diabetes in the different states in the United States. Actually, I look at Minnesota and actually it's low, so that's great actually. Of course, type 1 diabetes is an autoimmune disease that was first understood as a T-cell driven disease due to prominence of T-cells in insulitis. It starts with a spark or a trigger actually that leads to the initiation of the process that can be related to genetic predisposition, to environmental factors, perhaps the diet or infection with different viruses, or shaping of the T and B cell repertoires, and trigger that actually initiates some beta cell damage, islet damage, and is known, this is from biopsies from humans with type 1 diabetes and corresponding control. This is done by Peter Butler in the early 2000s, showing that actually apoptosis is increasing beta cells in type 1 diabetics. That leads to an immune recruitment, activation, and subsequent more damage, leading to a decline of beta cell function, beta cell mass. There is more antigen presentation and activation of adaptive immune responses that keep increasing the death of the beta cells, and loss of first phase insulin response, and further decline on beta cell function that continues without any intervention to be actually leading to the disease course actually. So if there is any treatment with, for example, immunotherapy, there is a chance actually that this could be recovered and or partially or totally actually, and at least the disease will delay or disappear theoretically in the future. It's also important extracellular matrix preservation, and I'm going to say something about that. Just let me show you actually that the decrease in apoptosis actually leads to a decrease, a pronounced decrease in beta cell mass, actually, and beta cell area in these patients. So as I said, islet extracellular matrix is important actually, is a form of many different oligosaccharides and proteoglycans, although heparan sulfate seems to be one of the main ones actually identified to encapsulate the islets. This is a study by Corpus and colleagues in diabetes in 2013, showing, for example, in this whole mount of pancreas, and this is an islet from a mouse that is type 1 diabetic, showing actually staining for laminin with a panlaminin antibody in green, and showing in red CD45 immune cells invading and chewing actually the extracellular matrix of the islet and getting inside of the islet. And there is a correlation between the integrity of the peri-islet membrane extracellular matrix actually presence and how it disappears over time, increasing the number of CD45 cells in the islet. Exactly the same has been shown actually for not just for NOD mice, but also for human islet pancreas. For example, this is a comparison from Simonovich, a group of pancreatic sections from MPOT, from controls, or from type 1 diabetics, showing a decline in heparan sulfate staining in the human islets from this type 1 diabetic subject, and also proteoglycans such as Syndecan-1 also losing their staining almost there, suggesting that actually there is a correlation between the integrity of the extracellular matrix and the disease progression. So it has been suggested actually that perhaps what is happening is that immune cells produce heparinase actually that degrades the extracellular matrix in the vasculature, and that leads to leakage of the vascular barrier and invasion of the islets with immune cells that actually keep degrading the heparan sulfate in the outside of the cell of the beta cell, leading to the invasion of the cell with the immune cells in the surrounding area of the cell that produce cytokines, and that leads to a more decline in heparan sulfate and an increase in ROS and cell death. So there is this hypothesis in which if we increase the way to inhibit the heparinase action, we will not allow the immune cells to do extravasation into the inter-islet area, and then it will not induce an increase in ROS and no increase in apoptosis. There are multiple targeted experimental therapies right now ongoing in the whole world in terms of trying to decrease the incidence or ameliorate the type 1 diabetes, in the sense that actually they are affecting many different pathways from the immune cells, and you can see it's a very complex, in this drawing, actually a very complex effect, but also trying to protect the beta cells some way in which actually the immune cells induce a lower effect or damaging effect in the beta cell. So from antigen-specific oral insulin and others to DQ8 blockade, to anti-CD3, to CTLA4, anti-CD20 to eliminate B cells actually, or trying to make naive cells into Tregs, or actually blocking cytotoxic lymphocytes with ATG, etc. There are many ways actually in which these effects can be, these pathways can be affected, so in a way that actually the attack on the beta cell is decreased. But there is no molecule here that can protect the beta cells at the same time that induces immune modulation and increased tolerance against the beta cells that they are being protected. So we thought that perhaps Dextran sulfate, that is a semi-synthetic sulfated polysaccharide, could perhaps help on these matters. So why? Because Dextran sulfate actually is an analog of heparan sulfate, so it could help into the extracellular matrix in the islet. Indeed, actually had been shown that replaces proteoglycans that are shed from the endothelial cell surface on activation of endothelium. Is a cytoprotectant in organ transplantation. Increases the production of a very well-known growth factor in which we have worked for many years, as Jan said in the introduction, that is hepatocyte growth factor. Hepatocyte growth factor is known to protect beta cells from multiple damages, included cytokines, included many other damages. On the other hand, also dextran sulfate inhibits heparinase, so that way actually will allow the intracellular matrix to be intact. It inhibits also the complement pathways, inhibits endothelium damage induced by ischemia reperfusion, inhibits the adhesion of neutrophils to endothelium, and also it has been shown that is capable of decreasing the functional maturation of human dendritic cells, at least in vitro, leading to the decrease in cytokine production and a decrease in proliferation of T cells. So all of these properties of dextran sulfate, low molecular weight, makes this molecule really attractive in the sense of testing it for whether it could ameliorate type 1 diabetes. However, before to go there, actually nothing was known about dextran sulfate effects on pancreatic beta cells, whether actually will be also an agent that could protect beta cells from different types of damages. So the first experiments in which we worked for a few years is what to analyze the effects of dextran sulfate in beta cells per se. So for that, we used human islets from cadaveric donors, actually, in which we could put them in culture and treat them with different doses of dextran sulfate from 100 to 500 micromolar, then treated with cytokines as well, or the cytokines plus the dextran sulfate. Immediately we noticed actually that when we stain these human islet cells with insulin, here in red, or tunnel as a marker for cell death, and DAPI for the nuclei, we noticed that, of course, with cytokine treatment, there was an increase in nuclei that they were actually positive for beta cells that they were dying. Of course, when we treat them with dextran sulfate, actually, we saw a decline in the number of nuclei that they were positive for tunnel and insulin. And here is depicting 5 or 6 different, actually, human islet preps, showing that actually cytokines increase cell death, beta cell death, and thus decreased by dextran sulfate treatment. And by the way, if anybody wants to interrupt me, it's fine. I mean, on the way, I mean, if there are questions, it's fine with me. No problem at all. Then also we tested whether actually signaling by the cytokine was altered by dextran sulfate, and we noticed that actually all the signals that are cytokine mediated, such as, for example, phospho-STAT1, or very clearly NF-kappa B, or for example, inos, actually, that was really upregulated by cytokines in humans, was highly reduced by the dextran sulfate treatment, indicating that cytotoxic cytokine signaling was inhibited by this polysaccharide. Not only in whole islets, this is just an example. So what we did next is to determine whether actually this decline, for example, in phospho-STAT1 by dextran sulfate was also in beta cells, or it was in all the cell types in the islet. So for that, what we did is to get human islet cells, disperse them, go to the flow cytometer after staining with C-peptide antibody, and detect the C-peptide positive cells that they were also stained for phospho-STAT1. And we found a shift from cells that they were treated with cytokines. You can see the phospho-STAT1 upregulation here, a shift to the left actually in the peak when we treat them with dextran sulfate and cytokines, indicating that C-peptide positive cells have a decline in phospho-STAT1 positivity, meaning that the cytokines were not able to activate the signaling pathway in beta cells, in human islets, in the presence of dextran sulfate. Of course, the decline in inos actually led to also a decline in the amount of NO and nitric oxide that was formed in these human islets. Here you have it also by DAF-FM staining, showing that with cytokines and cytokines with dextran sulfate treatment, there is a shift to the left actually in the peaks. ROS formation, reactive oxygen species, in those humans was also declined by or decreased by dextran sulfate treatment in the presence of cytokines. But not only the treatment with cytokines that could be perhaps blocked actually by dextran sulfate, impairing the signaling extracellularly. What we found is that actually when we induce ER stress in these human islets with the use of tapsigargin, that causes actually beta cell death, you can see here, in different human preparations. When we treat them with dextran sulfate, actually, dextran sulfate was able to impair the tapsigargin-mediated damage in human beta cells and in a dose-dependent way. This was confirmed actually by Western blot analysis of a target of ER stress that is terminal and linked to beta cell apoptosis, that is CHOP upregulation. You can see tapsigargin led to the upregulation of CHOP, and the treatment with dextran sulfate decreased the expression of CHOP in whole islets, in human islets. So dextran sulfate is capable of protecting beta cells from cytokine-mediated death, cytokine signaling, and actually also is capable of decreasing NO formation, reactive oxygen species upregulation, and ER stress. All of these are candidates for initiating beta cell death, and some of them are culprits actually of initiation of type 1 diabetes. Important, actually, I mean, that I put this in here because it's important, and I'm not gonna continuously say this, but I mean, what we found with dextran sulfate actually didn't appear to happen, or it didn't happen, when instead of dextran sulfate, we used Dextran, so non-sulfated Dextran, meaning just the polysaccharide without any sulfation, is not capable of actually reducing the beta cell death induced by cytokine. You can see the same doses of Dextran, low molecular weight, compared with dextran sulfate. Actually, it didn't induce any beneficial effect in terms of beta cell survival in these environments, in this treatment, indicating that sulfation is important. After showing that dextran sulfate was a potent inhibitor of beta cell death induced by cytokines, we wondered whether actually the profile of genes that they were involved in inflammation in human islets treated with dextran sulfate was also changed in these islets treated with dextran sulfate as well. And we performed RNA-seq analysis of human islets treated with cytokines or human islets treated with dextran sulfate plus cytokines. We found by gene ontogeny analysis of the pathways that they were activated or repressed in these studies, we found that actually, of course, apoptosis pathway was highly repressed, NF-kappa-B signaling was highly decreased, interferon gamma as well, TNF-alpha as well, and many others such as chemokine signaling, etc. On the other hand, activation of pathways such as oxidative phosphorylation, pyruvate metabolism, mitochondrial function, etc., were upregulated, as I said, in the dextran sulfate plus cytokines, perhaps indicating that actually the mitochondrial activity and the function of the cell was protected actually with dextran sulfate from the damage induced by cytokines. And we will see that in a few minutes. These are just different hallmark plots, actually enrichment plots of different pathways here depicted here. For example, in the genes that they are activated or upregulated in the cytokine treatment. They are not as high in the cytokine DS treatment for a particular pathway such as inflammatory response, the cyclase for apoptosis, the cyclase for TNF-alpha signaling, and the cyclase for interferon gamma response. So all the pieces seem to come together as all the inflammatory signaling pathways induced by cytokines, as I showed you before in the Western blots, also at the gene level seem to be suppressed with dextran sulfate. When we looked at the chemokine genes, and this is a list of the chemokine genes, in different human preps treated with dextran sulfate or cytokines, or the combination, we found that actually many of these chemokine genes were clearly downregulated in the human treated with dextran sulfate, suggesting actually that perhaps the anti-inflammatory effects that this could have is because also the cytokine, the chemokine expression actually is downregulated in these human islets. Looking at one of them, for example, CXCL10, we found that actually a low dose of dextran sulfate, 100 micromolar, had effects on survival, but they were less than 500. It was able actually to decrease even the levels of CXCL10 release from human islets treated with cytokines to the media, indicating that actually, again, that the chemokine response is decreased in dextran sulfate treated human islets. As I mentioned before, there are several genes that they were upregulated in the mitochondrial function pathway. So the next step for us was to see whether actually those changes in the profile of mitochondrial genes were related to changes in mitochondrial function in the islets treated with cytokines on dextran sulfate. So we performed a seahorse analysis. The oxygen consumption rate was measured in these human islets treated with cytokines, here in green. In blue is the control. In red is the human islets treated with dextran sulfate alone, or in purple, the cytokine-treated human islets, but also treated with dextran sulfate. You can see that actually the basal oxygen consumption rate was partially recovered by dextran sulfate treatment. It is important to show that in general, the OCR, the oxygen consumption rate, was heavily diminished, actually, even in the presence of high glucose, or the different drugs to measure mitochondrial function. It was highly decreased compared with the dextran sulfate plus cytokine treatment. Indeed, quantitation of ATP production from this experiment showed that actually dextran sulfate plus, cytokines in the presence of dextran sulfate actually don't have a pronounced decrease as shown with cytokines alone treatment, suggesting that actually dextran sulfate recovers or protects human islets from losing their mitochondrial function. That has a correlation with beta function as well. And for measuring that, we performed islet perfusion assays. And this islet perfusion, basically, we just place human islets in a column, and we perfuse these islets with different solutions of glucose or different secretagogues that allows to measure different insulin secretion phases with these different glucose concentrations. This is performed, as Jan said, actually we have the human islet and adenovirus core as part of the Diabetes Research Center. That is the Einstein and Sinai Diabetes Research Center. That is a joint effort actually to have different cores on diabetes-related techniques. And in this case, in the Diabetes Research Center, we measure insulin secretion by perfusion analysis. And here you can see in green again, human islets treated with cytokines show a very blunted first phase insulin secretion and a completely flat second insulin secretion, second phase insulin secretion. When we treated with dextran sulfate plus cytokines, actually there was some recovery on the first phase insulin secretion and the second phase insulin secretion. And indeed, when we perform multiple human preps, we saw that actually the area under the curve, or the stimulation index actually, of the insulin secretion in humans treated with dextran sulfate and cytokine was similar to control and very different from cytokine treated islets, indicating that actually human islets protect beta cells from cytotoxicity in terms of death induced by cytokines, but also from the damage induced in the mitochondrial function, as well the damage induced in the beta cell function, protecting it to have a normal function per se. So with these beneficial effects on beta cells, we aim at determining the effects of dextran sulfate in vivo in type 1 diabetes mouse models. In particular, actually, what we did is to get dextran sulfate, you can buy from Sigma, low molecular weight. We are using always between 5 and 10 kilodalton. And we did daily IP injections in NOD, female NOD mice, actually, and that is the mouse model of spontaneous autoimmune type 1 diabetes. And we treat pre-diabetic mice for 1 month or 1 month and then stop the treatment, or for 3 months, or we did actually even treatment of diabetic mice already there. and treat them for almost 3 months as well. In this case, I will talk later about what it means progressors or acute, actually. There are 2 different types of diabetic mice in NODs, actually. So anyway, it looks like. And then we measure after treatment glucose homeostasis. I mean, during the treatment, we measure glucose homeostasis. At the end, we measure beta cell homeostasis, insulitis, immune cell phenotype we perform, and also we obtain other organs to see the effect of dextran sulfate treatment in those other organs. So first, it was the pre-diabetic model in which we treat the pre-diabetic mice. They were non-diabetic. We treat them with dextran sulfates for 3 months, actually. And we found that when we use 10 milligrams per kilogram of dextran sulfate injection IP, there was a significant reduction from 70–75% of mice becoming diabetic in our colony in these 3 months to only 20% of them becoming diabetic. When we use a lower dose of dextran sulfate, that effect, although initially it seemed to be there, it seemed to disappear over time. So actually, a very reduced effect. So it is dose dependent, but 10 milligrams per kilogram definitely decrease the incidence of type 1 diabetes in these mice. And here you have the blood glucose levels on the individual mice. Plasma insulin levels. Before the treatment, we took some mice, sacrificed them at 8 or 9 weeks of age. Actually, we measure plasma insulin levels, and then we measure also the plasma insulin levels of the mice treated with saline and dextran sulfate. You can see the decline in plasma insulin levels that actually is maintained. The insulin levels are maintained by dextran sulfate treatment in these mice. Interestingly, we need to do the treatment daily on these mice to maintain these protection against the development of diabetes in NOD mice. Because when we did one treatment for just 4 weeks, actually, we stopped the treatment, and then we kept treating them for a period of time, we didn't see an improvement in the number of mice that became diabetic in this cohort of mice, indicating that the dextran sulfate present is required actually for ameliorating type 1 diabetes in NOD mice. The other effects that we see in the pancreas, we perform beta cell homeostasis, is that the beta cell mass, that this is the beta cell mass in mice before treatment. Actually, this is a cohort of mice that we sacrifice, as I said, at 8–9 weeks. After the treatment with saline for several months, there is a decline in beta cell mass, characteristic of these mice, and that was not present actually, or in some mice, but minimal number compared with the ones that they maintain a similar amount of beta cell mass in their pancreas. When in this pancreata we measure tunnel and to analyze beta cell death with insulin staining, we found that actually apoptosis was highly increased in the saline treated mice, and that was reduced actually in the DS treated mice as well. So DS and dextran sulfate decreases cytokine-induced cell death in vitro in human islets, also in mouse islets, but also decreases apoptosis or beta cell death in beta cells actually after treatment for 3 months in NOD mice. As I said before, I mean, I'm not going to say this frequently, but I mean, dextran, we have cohorts of mice in which we use dextran instead of dextran sulfate to see the effect of sulfation, and we found that dextran actually didn't improve the percent of mice that became euglycemic or maintained euglycemia over time, and also the tunnel positivity in beta cell was not different in these mice treated with dextran compared with saline control. Insulitis was reduced in the mice treated with dextran sulfate, and there were many more islets that they don't have insulitis or pre-insulitis. On the other hand, there was a decline in the numbers that actually have medium to severe insulitis. So insulitis scores were reduced. However, and interestingly, actually the effect that we see with dextran sulfate is more protection and protection against cytotoxic effects of cytokines, of immune cells, ER stress, etc. But we didn't see an effect on proliferation of beta cells by dextran sulfate treatment. So it's more of a way to preserve beta cells than to regenerate the beta cells that are still remaining in there. So that's an important point because, I mean, we'll talk about that later, but if we want a way to regenerate beta cells in type 1 diabetics that they lost a high percentage of beta cells, Dextran sulfate probably will not work in that particular setting, and it will require the addition of additional agents actually that can increase proliferation and regeneration of beta cells. As I said before, also extracellular matrix is important, so we measure by staining of alcian blue for heparan sulfate or heparan sulfate proteoglycan antibodies to detect whether actually the extracellular matrix was being protected in Dextran sulfate treated mice, these pancreatic sections from the previous mice I just showed you. And actually, you can see that the extracellular matrix is being preserved. As I mentioned before, it will be important perhaps to find an agent that can decrease heparinase activity, and we found that actually Dextran sulfate inhibits, as it has been previously described, heparinase activity. So the amount of heparan sulfate that is degraded by heparinase decreases when we use an increased amount of Dextran sulfate. And also in human animals, in vitro, actually, when we treat them with cytokines, we see a decline in heparan sulfate proteoglycan staining. You can see here, of course, we see a decline also in beta cells as well, or islet cells as well, and that's preserved by Dextran sulfate plus cytokine treatment in these human animals, indicating that actually Dextran sulfate is capable of inhibiting heparinase activity, and perhaps that way actually protect the extracellular matrix in the islets of mice or humans actually that they are treated with cytokines. So it has multiple effects actually in terms of cytoprotection of beta cells against cytokines induced cell death, protects mitochondrial function, protects beta cell function, protects beta cells in vivo in pre-diabetic NOD mice, protects or maintains actually the extracellular matrix in the islets. However, it doesn't increase beta cell proliferation. That's what a summary actually what I just said. So then we decided to go on one step further and make things a little bit more complicated. So we got mice that they were diabetic. So we wait for them to become diabetic. We measure glucose every other day actually for several weeks until we saw mice that they became diabetic. Saying diabetic is 250 milligrams per deciliter. And then we start treating them at that point with saline or Dextran sulfate daily for 10 weeks. At the end of the 10-week treatment, you can see that actually only 40% of the diabetic mice remain diabetic, while 100% of the diabetic mice stay diabetic. So there's a significant reduction actually in the number of mice that became diabetic after Dextran sulfate treatment, indicating that at least in a large percentage of diabetic mice, there is some reversal of the diabetes in these mice. It's important, just you're going to see it later again. These are what they are called progressors, actually, in terms of mice that they progress to diabetes slowly. Okay? And this is going to become important. In terms of plasma insulin, we see actually an increase in plasma insulin levels. Of course, in these mice treated with Dextran sulfate, they are not becoming diabetic. We see the islets kind of intact in the pancreata, actually, although surrounded by massive immune cells actually, or cells actually around it. Of course, in the saline treated mice, the beta cells have disappeared. There are cells all around it. When we do a staining for for B cells with B220 antibody or T cells with anti-CD3 antibody, we find that the saline treated mice, the islets are surrounded by a massive amount of T cells and actually B cells as well. However, in the Dextran sulfate treated islets, we find also the T cells and B cells all around, although they are not inside of the islet. So that's an interesting point, actually. So they're still there around it, but kind of impairing the perhaps the stabilization or the action on the beta cells diminished in a way that actually the beta cells are surviving longer. Beta cell mass, of course, is increased in this situation. This is all in terms of beta cells. What about the immune system? What is the effect of Dextran sulfate on immune cells? Well, we find that actually when we get splenocytes and we do gene expression analysis of many different genes involved in activation, maturation, and phenotype of different cell types, actually we find changes in the Dextran sulfate treated mice in a way that actually, for example, interferon gamma expression is decreased, IL-10 is increased, phospho-3 is increased, among many others. This is just an indication that probably immune modulation is happening with Dextran sulfate. When we use Dextran, that effect disappears completely. There is no effect actually on any of the genes tested. So is Dextran sulfate, again, the drug that is causing all this modulation of the immune system? When we look at the specific immune cell types, actually, we look at CD4, CD8s, and we look at interferon gamma, Th1 cells. We found that actually Dextran sulfate treatment in vivo was capable of decreasing the number of interferon gamma positive CD8 cells in pancreatic lymph nodes or splenocytes situation, and the same happened with interferon gamma CD4 positive cells. When we use Dextran instead of Dextran sulfate, we didn't see a change in the activation of these different CD4 and CD8 cell types. When we looked at PD-1 as a program cell death protein 1 that is very important for suppressing T cell activation, we found that actually CD4s and CD8, again, in pancreatic lymph nodes and splenocytes, have an increase actually in the number of cells that they express PD-1 compared with the ones that they don't express it. So indicating that actually this marker of tolerance, self-tolerance, was increased in T cells. We have recently also analyzed myeloid cells and found that actually PD-L1 is also upregulated in those cells, suggesting that perhaps the PD-1, PD-L1 signaling pathway is modulated by Dextran sulfate some way. We are trying to analyze what is the mechanism for that. Interestingly, when the mice that they were pre-diabetic were treated with Dextran sulfate or saline, but at the same time were treated with an antibody, anti-PD-L1, all the mice became diabetic in saline-treated mice, and almost all of them that they were treated with Dextran sulfate became diabetic as well, indicating that blocking PD-L1 with an antibody completely blunted the effect of Dextran sulfate to ameliorate diabetes in diabetic NOD mice. On the other hand, so decreases the activation of Dextran sulfate decreases the activation of T cells, CD4, CD8s, increases the self-tolerant marker PD-1 and PD-L1. I can tell you that too. And looking at regulatory T cells, we find that actually Dextran sulfate is capable of increasing the number of phospho-3 CD25 positive CD4 cells and phospho-3 in both in pancreatic lymph nodes and spleen as well. So increases the number of Tregs, and actually not only increases the number of Tregs, this is a complicated figure, but I'm going to summarize it very easily, that the suppressive capacity of Tregs is also increased. These are in vitro assays in which Tregs treated with DS are co-cultured with activated T cells, and you can measure proliferation without any Treg, just T cells. Actually, you have around 60% of cells non-proliferating, but 40% proliferating. As soon as you start adding more Tregs to the activated T cells, the proliferation starts declining because there is suppression, and actually you need 1 to 4 to show a clear decline actually on the proliferation of these T cells. However, in Tregs that they are treated with Dextran sulfate, that decrease in proliferation is already observed into 1 and 216. So it's like a very potent suppressive capacity of Tregs treated with DS on activated T cells. I will go fast with this one, saying that actually we also measure whether actually Dextran sulfate was causing any effect on intestinal inflammation, because Dextran sulfate has been shown to, in the drinking water, has been shown to increase inflammation in the intestine. However, just IP injection with saline, we didn't see any alteration of any of the markers of inflammation in the intestine. Interestingly, this is studies are showing also that the effect of Dextran sulfate seems to be mostly related to the type 1 diabetes effects, and it's not just a general immunosuppressive effect actually, because when we analyze sialitis in these NOD mice treated with Dextran sulfate, we didn't see any change in the sialitis score actually in the salivary glands of these mice, indicating that it doesn't have an effect in that sense. And actually, we also performed septic-shock with LPS injection, and we didn't find any significant difference in survival or cytokine profile on these mice, indicating that the mice are capable of responding to effects actually induced by inflammation that they are not related to diabetes or autoimmunity that is not related to diabetes. So it's kind of a specific effect on type 1 diabetes. An important point here is that actually the effect happens in what is called progressive diabetes in mice, and these mice go slowly from non-diabetic to diabetic and still go slowly. In those mice, actually, the Dextran sulfate has an effect. However, what they are called acute type 1 diabetic mice, meaning they become diabetic very quickly, actually, from being glycemic to very highly hyperglycemic in just 1 week. In those, actually, the treatment with Dextran sulfate doesn't work, and it's an important point. And this all comes actually as previously described by Clayton Matthews and Mark Atkinson, showing that actually in their cohorts of mice, and they have many mice actually over the years, they can see that there are two different cohorts of mice. Ones that they are acute NOD mice that they become diabetic very rapidly in 1 week, while the other ones, they become very progressively diabetic. Interestingly, the treatment with ATG and CSF of these mice, that is a combination therapy that will reduce the reverse diabetes, that occurs only significantly in mice that they are progressive to the disease, meaning with this slow progression to hyperglycemia. In acute mice that they become diabetic very rapidly, actually there is no effect. This is important because probably type 1 diabetes is very different in very different human subjects, and that would indicate why some of the immune therapy actually is working in some individuals but not in others actually. So it will complicate a little bit the effect of the drugs that we are going to be testing. Also, it could be that actually in the acute one, beta cell death actually is very rapidly occurring, and actually there is almost no beta cells at the end, and that's why there is no effect of immunotherapy or actually protecting beta cells, because the number of beta cells that they are at the end is very limited. So Dextran sulfate protects human beta cells against cytotoxicity induced by cytokines, GI stress. Dextran sulfate reduces the mitochondrial and functional damage induced by pro-inflammatory cytokines. Dextran sulfate reduces T cell activation and increases Tregs, and prevents diabetes development and reverses early onset diabetes in NOD mice. However, it has to be daily administration, and known optimal dosage and routes. It's unknown the effects in humanized mouse model of type 1 diabetes. It's unknown the effects in human immune cells except for human dendritic cells. And it doesn't have any beta cell regenerative action in type 1 diabetes. So how can we induce beta cell regeneration? And this is more related to what Yang talked about during the introduction, that is the identification of a specific small molecule doing high-throughput chemical screen, actually, of agents, more than 100,000, that they were able to increase beta cell proliferation. It was a collaborative effort led by Andrew Stewart and Pen One in his lab, with the contribution from the labs of Roberto Sanchez, the Don Scott, my own lab for these studies. Basically, it used a high-throughput chemical screen to identify agents that they could increase beta cell proliferation. Of those, actually, the one that it was the most, the agent that actually was able to increase beta cell proliferation most potently was Harmine. Harmine that increased not just rat beta cell proliferation profoundly, it also increased human beta cell proliferation as well in a marked way. What is Harmine? Harmine is an alkaloid from plants such as harmal or ayahuasca is a psychoactive compound with antidepressant properties, have been used in the past. Selectively binds to and inhibits the T1A, or the dual specificity tyrosine kinase regulated kinase, one that participates in the regulation of the nuclear factor of activated T cells, or in fact, actually, and also inhibits monoamine oxidases and CDC-like kinases. Also works or activates PPR gamma in adipose tissue, increasing the glucose sensitivity in diabetic DBTB mice. So it has effects in the brain, but also is a potent inhibitor of T1A, and it has effects in peripheral tissues to increase insulin sensitivity and increase blood glucose levels. The way it works actually is that calcium gets into the cell, calcineurin gets activated. That what it does is a phosphatase that eliminates phosphate from the transcription factor called NFACT. When NFACT is not phosphorylated, then it goes to the nucleus and binds to many different promoters of genes that are involved in proliferation. These are cell cycle genes. T1A, what it does is actually is not permitting to get constant proliferation. So what it does is phosphorylates NFACT and maintains NFACT into the cytoplasm, so there is no activation of cell cycle genes and the cells are at rest. However, Harmine, what it does is actually block T1A, so now when it's dephosphorylated, it remains in the nucleus and it doesn't go back because it doesn't get phosphorylated by T1A because it's inhibited. The increase in proliferation induced by Harmine that inhibits T1A, not only, as I said, increases proliferation, but actually it doesn't affect insulin secretion. Increases actually the expression of beta cell genes such as PDX1 and AGX6.1, and actually not just at the level of genes, such as MAF-A as well, but at the protein level. So that's notion that actually increasing proliferation induced by agents leads to the differentiation of beta cells, it doesn't happen with Harmine, because Harmine induces proliferation and at the same time increases the expression of beta cell markers and increases and maintains insulin secretion. So it doesn't lead to a beta cell, a decrease in the beta cell function. Not only increases proliferation in vitro, but also it does it in vivo. So these are human islets transplanted under the kidney capsule in immunosuppressed mice, and this is the graft actually showing Ki67 staining, so proliferation is markedly decreased. But also actually improves glucose homeostasis in diabetic mice, immunosuppressed mice, treated with human islets, and then treated daily with Harmine for 3 weeks. And we can see that actually blood glucose levels go down and they're maintained low until we perform ureterectomy and the glucose goes up again. So indicating that actually it's capable of improving glucose homeostasis in transplanted mice with human islets. And that correlates also with a better glucose tolerance in these mice. So this is the effect of Harmine. So this is what also Jan was saying at the beginning, that a proliferation of insulin-producing cells, or beta cells, actually is rapid at the beginning in the neonatal ages, and then it declines in the childhood and adolescence, and then when you are an adult, your proliferation of beta cells is very limited. So here's what we found in vitro, so it's very similar. When we add Harmine, we can reach levels similar to what you get actually in neonatal age, but that will not be enough probably if you want to replenish 80%, 70%, 90% of your beta cell mass if you are type 1 diabetic. So we need to think of ways that we can further enhance human beta cell proliferation, combining Harmine with other agents that stimulate complementary signaling pathways in beta cells. And this was published in Science Translational Medicine this year, in which, again, the effort by multiple labs led by Andrew Stewart combined GLP-1 receptor agonists together with T1 inhibitors such as Harmine and tested the effect of that combination. And we found that actually the GLP-1 receptor agonists with Harmine synergized to increase proliferation now to a levels actually around 5%. That is a much larger effect actually with increase the numbers. Indeed, actually, when in vitro we measured the number of beta cells that they were before treatment and after treatment for 3 days with GLP-1 and Harmine, there was like a 20 to 30% increase in the number of cells actually at that point. The effect not only happens with Harmine high dose, but also when you use a lower dose of Harmine that almost doesn't induce proliferation, when you combine it with GLP-1 receptor agonists, you see a full activation, a full increase in proliferation. This is important because if we can reduce the dose of harmine, now the side effects will be reduced and the psychoactive effects, for example, disappear because the dose will be so small that actually will have limited effects in that sense. But if we add GLP-1 on top of that, now we have the same proliferation that we got with 10 micromolar of harmine. The effect of synergistic effect is not only just with GLP-1 and extending 4, it's also with all the different drugs that they are being used to treat diabetic patients, such as liraglutide, lisirnatide, etc., semaglutide, all the drugs. And actually not only works in human islets by itself, but also in type 2 or islets from type 2 diabetic donors, actually. And you can see that actually proliferation is also increasing in these islets from diabetic donors, and function in terms of insulin secretion and expression of different genes is also increased in this situation. And as I previously said, actually harmine, in this case harmine plus exenatide, and in combination of low dose of harmine and exenatide improves glucose homeostasis in diabetic mice transplanted with human islets. And human plasma insulin is increased as well, and glucose tolerance is increased. However, does it lead to an increase in beta cell mass, beta cell volume? For that, we went to and use a technique that is called IDISCO. That is a method that clarifies the tissue, and it was done by Carolina Roselot in my lab and Jan Suili with Sarah Stanley and Alexandra Armanson help. And actually, basically, what we do is we get human islets, transplant them, infuse the mice with the drugs, in this case, harmine plus exenatide. We get the kidney, we completely clear the kidney in a way that becomes transparent. Now we can add antibodies to label different tissues, in this case, for example, anti-insulin antibody or a smooth muscle actin, and we can see the transplant actually on top of the kidney in a 3D image using light sheet microscopy. So this is one of the transplants, for example. So once it becomes transparent, actually we can stain them with different antibodies, and you can see actually the graft with insulin in green on top of the kidney, and then the kidney labeled with a smooth muscle actin. Now using IMARES software allows you to calculate the volume of all these cells. So now you can calculate beta cell mass, beta cell volume in 3D and compare different treatments. So that's what we did. We did 12 weeks treatment with pumps with harmine and exenatide, and we find that 3 months after the treatment with harmine plus exenatide, you can see very obviously that actually the graft of the vehicle-treated mice is small. However, the graft of the harmine plus exenatide treated mice actually has increased enormously. And when we do multiple, up to 7 different human islet transplanted with, mice transplanted with human islets, we find that harmine increases like 2-fold the mass. However, the harmine plus exenatide increases the mass of human beta cells by 7-fold in 3 months. So that now becomes closer to what we want in terms of regeneration. So we can say that the combination of harmine plus exenatide remarkably increases human beta cell mass in vivo in human islets transplanted in glycemic immunodeficient mice. However, we don't know whether the increase occurs only in diabetic conditions as well. This is euglycemic mice. Whether this increase in islet mass occurs in other islet and not islet cell types, and whether harmine and any other harmine analog can be targeted to the beta cell, and that's areas in which we are working. And I would like to finish, I'm taking too much time, sorry about that, is my lab, definitely the studies on Dextran sulfate done by Brian Liu and Rondi Kang, the studies in all the harmine story together with Andrew Stewart is by Carolina Roselot and Jan Suili, and different collaborators actually, like Dirk Homan, as I mentioned before, and funding from different ages, and of course thanks to the human islet donors for providing us with the human islets to do all these studies. And thank you very much for your attention. Sorry, it took a little bit long.
Thank you, Adolfo. That was great. We have some time for a few questions. So,
Leo Furt here. That was a terrific talk, Adolfo. Heparan sulfate has been near and dear to me and a number of researchers in the department, Jim McCarthy and more and other chondroitin sulfate, and Andy Nelson and some others relative to tumor metastasis. I'd like to just sort of bring some of the importance of this to the plague we're dealing with right now in COVID. Mm-hmm. And I'm sure you know this, but I would imagine many on the Zoom. But heparan sulfate is a synergizer for binding of COVID-19 to the ACE2 receptor. And there's been historical work to show that heparin or heparan sulfate will actually decrease, is involved in the infection of herpes viruses. And actually, a few decades ago, Hank Balfour in the department, we did some studies. I don't know if we published it or not, but we showed that heparin binding peptides added to HIV cell culture model, in fact, decrease the infection with HIV. And there are anecdotes with other coronaviruses and heparin, and also a number of other viruses. So this is extremely interesting, and it's very similar in the models that are projected are very similar to yours in the role of leukocytes and other cells, cytokine, the beginning of the cytokine storm. And as many are probably aware, really ill patients are getting heparin as part of the regular anticoagulation therapy, but there's another salutary effect of that. And, it's quite exciting. Also, Jim McCarthy, who I don't know if he's on the Zoom because there's so many people you can't. He made a pioneering discovery 3 or so decades ago to show that in addition to integrins binding to ECM molecules and the like, there were heparan binding and chondroitin binding domains of adhesion molecules that were critically important in these interactions. So your work is very, very exciting. I really love the work on the expansion of the islet mass. And I actually suggested to our transplanter many years ago for islet transplants that they think about using, ECM molecules a cocktail of ECM molecules to encapsulate or facilitate take of the islets in transplantation, because as you know, there's a great deal of islet death associated with that. So thank you for the very stimulating talk.
Thank you. Actually, that's fantastic. So what you said is completely right. So in the 80s, actually, Dextran sulfate was used initially for HIV patients, especially in Europe, actually in Germany, I think. And it was with some success, often not complete, because it was blocking, I think it was CD4 actually, and the entry of the virus to the immune cells, and that way actually prevented kind of temporarily the effects of the virus. I didn't know about the COVID-19. I think that's a very interesting story. Sorry. And definitely, I mean, I agree with islet transplantation and Dextran sulfate actually is being used, is being patented. Actually, it's patented in Sweden right now in a company to treat human islet transplants at the same time with Dextran sulfate to block IV-MIR response and the complement, so there is more survival of the islets actually that they are engrafted in the patient. So yeah, you're completely right. Everything you said.
Hi, Adolfo. This is Jim McCarthy.
Hi.
3 decades Leo. I mean, oh my God.
Yeah.
It's been a while. I did that when I was 5. So I agree. It was very interesting. And my question is more basic in terms of heparan sulfate. When you add antibodies, and I had this whole thing in a chat, but I didn't realize I could just join in. So your antibodies against heparan sulfate, what are those against? Are they against the glycosaminoglycan, or I think you said syndican is on the surface, or what are those antibodies recognizing? Is that
recognize the protein? I'm guessing. I mean, to tell you the truth, I'm not 100% sure. But I mean, in the ones that I saw from Simeonovich effects, actually, is the an antibody against Sindecan one, for example.
Against the core. So, okay, so my question is about the heparan sulfate, and so there's an older literature which talks about that if you add sodium chlorate to cultures, you can decrease the sulfation of the GAG, even though the GAG would still be there. And my question is, would you predict that chlorate would do the same thing? I guess I interpret your results as meaning that that heparan sulfate might be sequestering growth factors or chemokines or whatever, and when it starts to disappear, you start to get bioactive milieu, which is causing lymphocytes to come in and changing the beta cells and so on. So I guess just with your beta cell cultures, would it make sense to add chlorate to see if that causes the same thing? That would let you go after what kind of growth factors you might be thinking about.
Exactly. So that's a great point. Actually, we know, for example, that dextran sulfate binds to interferon gamma, many other cytokines as well.
Exactly.
And it has to happen through the sulfation. If it's not sulfated, the binding doesn't occur. So it is important there. However, I don't think it's just, dextran sulfate or any sulfated polysaccharide, probably what it has is multiple effects, not just binding cytokines and preventing them from binding to the receptor. It's also that is inducing the activation of other growth factors, for example.
Absolutely. Yep.
It's well known that dextran sulfate binds to AGF,
absolutely,
factor, and maintains AGF in the circulation without being degraded.
Right.
And actually favors binding to the receptor as well, increasing signaling. So some factors are favored on their actions, some factors are blocked on their actions. It is complicated because if you look at, right?
You're preaching to the choir, Adolfo. They are very complicated. It would give you a way to ask questions about the sulfation part of the equation that you're interested in.
It's very important, but so now we are pinpointing the role of hepatocyte growth factor on all this story, and we see that if we block, for example, the receptor with an inhibitor, dextran sulfate effects disappear. So that would suggest that actually the preservation of hepatocyte growth factor binding to the receptor is essential actually for the effect.
Right.
Not just inhibiting interferon gamma activity, probably also if you block the pro-survival actions of hepatocyte growth factor or even the immune modulation by hepatocyte growth factor, then lose most of the activity. So that's where we are going actually, because as you said, it's complicated. What signals are being blocked, we don't know.
Well, it's very interesting. Thank you for your presentation.
Thank you for your question.
And with that, we do have to sign off this morning. Thank you very much for joining us today.